We're going to look at multiple alleles today as another type of inheritance that some traits use this way to get passed on from generation to generation. Specifically, I want you to notice the picture because this is going to be very important when we're working some Punnett square problems in a few minutes. We mentioned blood type already. The AB blood type was an example of having codominance, so you might want to jot that down or remember that one. But in general, blood typing is an example of multiple alleles. And what that means is that if you look at the human population, there's more than two alleles for this particular trait that exist in the population. I want you to notice an allele, we have I with an A. That's one potential allele. We have I with a B is another potential allele. And we have little I. So there are three different alleles, and so the combinations of those, we have 1, 2, 3, 4, 5, 6. There are 6 different combinations, and so we have 4 different types of blood possible. So this is an example of multiple alleles, and we're going to do a few examples here of how you would do some Punnett squares when you have multiple alleles. So for our first one, we have 2 type AB parents and they have a child. So this is where you're going to want to reference that chart that was just on the previous screen. An AB blood type is going to have, and I'll just go back, the AB blood type, you're wanting to find the genotype. So this would be the blood for the genotype of an AB parent. Alright, so we have I with an A and I with a B. And it says we have 2 type ABs, so we're going to do that again. So that's our starting point, that's our parents. We have the genotypes of the parents, and we are going to break these apart because when these parents have children of their own, they're only going to be able to pass on one of these letters. And just like we have been in the habit of doing, please make sure that if a box like the one I'm in right now contains 2 different letters, that you do alphabetize them, put them in order. So we have a few different combinations here of potential blood types in our children. So we have a Punnett square. So what are the expected phenotype and genotype ratios of our offspring? Well, let's take a look at our phenotypes. We have A blood possible, B blood. These are just in the human population. These are what could potentially exist. So if we take a look at our boxes, we have an A and an A, so that's going to be an A blood type here. We have A with B, so that's an AB blood type. Another AB, so there's an AB blood, and 2 B's is a B blood type. So O doesn't show up here at all, so 0% chance of having type O blood. But if these 2 parents were to have offspring, there's a 25% chance of having type A, 1 out of 4 boxes. Type B is also 25% chance because 1 out of our 4 boxes showed that. And AB blood is 50%. There were 2 out of 4 boxes there. So that takes care of the phenotypes. Now the genotypes are what the actual letter combinations are. And there were 6 of them that I mentioned on the previous screen. I'm not going to write all 6 of them down. So we're going to go through our boxes one by one. We have A, I with an A, I with an A. And take a look at all 4 of our boxes and how many times does that particular AA combination show up. And it is just the 1 time, just the 1 out of 4 boxes. We also have IA and then an IB combination for our AB blood, and that one shows up 2 boxes out of 4. 2 out of 4, 50%. And then lastly, we have the IB, and that one only shows up just the 1 time, so 1/4 or 25. Just a 25. Let's make that look nice. So we have a 25% chance there. So that would be how we would use that chart and look at the blood types of a parent. Let's do one more. We have a type O parent, They have a child with a heterozygous type B. Alright, so we have two different types of blood than we had in the previous example. We have type O, and then we have heterozygous type B. So I'm going to quickly go through back here. Type O blood, there's only one genotype for type O, and that's 2 lowercase i's. And then for heterozygous type B, I want you to notice that there's 2 type B blood on here, and that is there's only one of them though that's heterozygous. Heterozygous still means different, so we have one with a B letter and one that does not have a letter. That's heterozygous because they're different, whereas this capital B, that's a homozygous combination. They're both the same. So when you're doing the Punnett square here, our type O parent is just the II. Heterozygous means different, so we want to have one with a letter and one without. So that would be our 2 parents. We have our genotypes. So now for our Punnett square, we're going to separate our letters. When our parents are making their individual gametes, we want to make sure that they only pass on one piece of information. And we fill out our boxes like we always have. Now, because we have a capital letter here with a recessive letter in this box being paired together, we want to put the capital first and the recessive second. So we're going to do that in both of those. And then down here, we're going to have little i's pairing up. So there's our Punnett square. So now the question is, at the hospital, they're handed their new baby who has type A blood. Is this their child, and explain why or why not. So I want you to take a look. If these 2 parents were to have a child, and these are all of the zygote options right here, so they can't have any other combination except for what's here in our Punnett square. We have an I with a B and a little I, another IB little I. So both of these show what type of blood. Hopefully you said type B blood. And then we have the little I and little I combination, and both of those show type O. So if I was this particular mom with a baby who had type A, and I would say, no, that is not my child, and my explanation would be that my husband and I do not have the potential of having a type A child. That's not a combination that either one of us could contribute. So definitely not our child. This is how they used to do blood typing before we had DNA testing. They could blood type a child with the parents and see if there was maybe a mix-up. We want to go one slide further here. We don't have a Punnett square for this one, but I want to mention just another type of inheritance. There's really not a great way to do a Punnett square for this. It's going to take almost like a supercomputer to try and figure some of these out. This one's called polygenic inheritance. And then especially in humans, we definitely have multiple genes that code for the same trait. So whenever we have a polygenic inheritance, that means that we have 2 or more genes that are going to contribute to a single phenotype. In this case, we're looking at how light or dark your skin might be, and that's definitely polygenic inheritance. Height is another one that's polygenic. You have at least 2, in this case for color, we have 1, 2, 3, 4, 5, 6, 7 possible different locations for positioning of these, and depending on how many of them you get or don't get for skin color is going to create very light skin all the way to very dark colored skin. So that's polygenic inheritance, and it just increases the amount of variety that we have. That's why height works very similar to this. You have some people that are way up here at, like, maybe 8 foot, 9 foot, and you have some that are very, very short. But then you also have this whole spectrum in between, kind of an average height there in the middle would have the most people, and so on either side. So polygenic inheritance we won't be doing any Punnett squares for, but I do know that it is mentioned on your test, and that you do need to know at least what polygenic inheritance is.